Okay, so here we go on chapter 10. We had a little bit of an intro before. Let me make sure we're recording. Great, great. Based on information we've already discussed in multiple other chapters, we just need to make sure it's replicated consistently. A lot of the mutations that we talk about, we used to think came from mutagens, but they often come from the polymerase just replicating 3 billion base pairs. It's going, our nucleotides, not a base pair until it makes its copy, right? At base pairs across the strand, nucleotides, what it's making. It's making that polynucleotide strand and making 3 billion replication units means that even if it's really great at its job, it's going to make a mutation every once in a while. Okay. So we're going to discuss this process in chapter 10. So it needs to be reproduced. It needs to have a template, and it can do it in different ways. It was proposed by Watson and Crick shortly following, just a few months after their proposal for the structure of DNA. It was proposed that the replication is going to serve as a template in the process of replication. And so these base pairing rules, let's see, green. This strand becomes unwound, and these empty hydrogen bonds are what determine what's going to be put in that place. Because a C can only bind to a G, the nucleotide will be put there even though the strand isn't synthesized yet. And then the polymerase will see that the let's say a C is right here next to the G, and an A is next to that T on the template strand. That new strand will have the nucleotides there, and then polymerase will catalyze the backbone. And that's basically all of chapter 2 in a nutshell. There we go, we're done. Anyway, so it can be done in different ways, and the process to discover which way was correct is the first of your chapter 10 with the different experiments, just like we had experiments at the start of the last chapter too. So it's proposed to be semi-conservative, and that's where it's going to use the complementary rules, or the Chargaff's rules, to insert from that template strand the new nucleotides that will become the daughter strand. So it has to have the unwinding so that it can access that template. And this was proposed publication-wise. They scooped this idea as soon as they put the structure out there, and that it bound by hydrogen bonds. So the three different, besides semi-conservative, it is semi-conservative, but what was proposed at the time and the possibilities was it could be conservative, it could be semi-conservative, or it could be dispersive. So let's start with conservative, okay? So conservative is you have one entire strand and you're going to make one entire daughter strand. So in my head, it would be like an enzyme that's like a tube, and somehow is going over the double helix, not opening the double helix, but going over it and somehow reading the bases that are inside and protected. It's a little bit harder for it to see it chemically though, because it's wrapped up in the double helix. But they don't know all these molecular details. I'm putting that kind of scenario. They might have had an idea, and there might have been researchers thinking it was conservative at the time. Semi-conservative is what we just presented on the last one, and that's where it pulls apart the parental strand, and then it's used when it's pulled apart with the empty base pair hydrogen bond sticking into solution as the template for the daughter strand. That's semi-conservative. So when you draw that out, you're going to have within one particular double helix, half of it's going to be parental and half of it's going to be the newly synthesized daughter strand. And then another possibility is you have chunks. So if you look right here, and then like right here, and then like right here, the colors are switching. So it's like you have a chunk of blue, and then you have the chunk of pink. And then you have a chunk of pink, and then you have a chunk of blue. So it's that process would be something in the cell that chews up the DNA and then makes a copy, then reassembles it. Which is possible. It's just, if you think about all these models, I'd have to say this seems the most simple once they knew about the hydrogen bond nature of the bases. And so there's a saying in science called keep it simple, in science and everything, keep it simple, stupid, and this really fits that, but it's also energetically favorable to keep that in mind. And so like chopping it up, you might have problems with damage, a lot of problems with damage. And so you have to keep in mind the genetic continuity, and you don't want to have that much mutation. And so you might worry about that might be a problem with that model. But that doesn't mean it shouldn't be pursued. So usually when experiments are designed, they're trying to test one model against another model, you know? And so, for instance, the conservative, it doesn't look like it would cause a lot of damage. It's possible they don't know the structures of different enzymes. And even now, when we do know the structures of different enzymes, I would still say it might be possible, you know, if we didn't know how it worked. We do know how it works, so we don't we don't say that. But so a lot of these first experiments were trying to compare the different possibilities to eventually get at if it is semi-conservative. So the Messelson-Stahl experiment is one that you need to know that looks at, you know, do you have a pink and blue strand made? Or is it conservative and you have a completely double helix of pink and a completely double helix of blue? Or completely, using the correct words, a complete double helix of a parental strand and a complete double helix of the new daughter, the nascent daughter strands, versus semi-conservative. And so they showed that conclusively that there's semi-conservative nature of the mechanism of replication. And they worked in bacteria. And they did this by feeding it a label of nitrogen. If you remember, the bases are called nitrogenous bases because not only do they have nitrogen on the edges in the hydrogen bond region of the base groups, but they all also have a nitrogen facing down to where they connect to the sugar. And so if the cell is going to metabolize and make that sugar to get ready for replication, then it's real dependent on the media you feed it. And so what the researchers did is they either fed it normal nitrogen, or they fed it a heavy nitrogen called N15 that has, let's see, Brenna, you're in chemistry. In just 5 years in this job, I've already forgotten a lot. So N15, if it's a heavy isotope, means that it has an extra proton, probably. Electron? Proton makes it heavier because it's in the nucleus. Electrons don't add much to the weight, but electrons add to the radioactivity. So it might be, anyway, it's whatever that atomic number up there without switching it to a new element. Yeah. Yep. So it's a radioisotope. It's heavier and it's physically heavier. It's not one that's highly energetic, which I think is more the electron, the gamma rays and stuff like that. This is one, we're not going to see it on an autoradiograph. What's going to happen in effect instead is because it's one molecular weight unit heavier across the entire DNA strand, your whole DNA strand is going to be like, boom, a little bit heavier in that chair. Okay, so they can see based on what types of, when they feed it that radioisotope and what they extract out at different time points, they can see where the heaviness is. Okay, so they go through rounds of growing the E. coli in heavy or normal N14. Yes, tell me. Neutrons. Oh, proton, neutron in the nucleus. Okay, neutrons. Yeah, because protons and electrons would change like the chemical properties, but neutrons are just like, who cares? Because they're neutral, so right. So it's heavier without changing the properties of it. Zoom people, it's a neutron. You're welcome. Anyway, so to be able to tell, you know, because it's a chemical, so we can't just watch like Dr. Howard sit in the chair and be like, oh, there's the quarantine 15/40. Instead, what you're going to do is a way to tell macromolecules if they're heavier is to spin them. And if you, when you spin them in a run, if they're heavier than another molecule, they will beat the other molecule and they'll be heavier. They'll go to the bottom of the tube faster. If they're lighter weight, they'll be towards the top. And so this is called sedimentation equilibrium centrifugation, or SEEK. Do you ever read a paper and SEEK is everywhere? That's what they're talking about. And they're just spinning the crap out of it. This is useful for purifying radioisotopes, which is why if you have one of these machines, you are on a list and you are watched. Not kidding. Anyway, so we don't have one. We have spin, but we don't have an ultracentrifugation, which is what it takes. E. coli is grown in that heavy media, and it's going to conveniently turn this into a color forest. Okay, so The heavy is going to be pink. And when you grow it in the heavy, it will result in a heavy labeled double helix because they're going through multiple rounds. This is their baseline. So they let it like, the replication cycle of the bacterium is less than an hour. It depends on the bacterium. So equalize around 20 minutes. It depends on if you're keeping it happy. But they're going to let that set in the heavy, like maybe overnight. They want to make sure all the DNA starts off from pink, 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 pink. Okay, everything's heavy. But then what they do to start the experiment is they're going to replace the media, which is really easy. You just not in this super speed thing over here, just in like a spin down, like on the bench. If you've ever used a spin down on it, just for like 10 minutes, you just they'll sink down by themselves if you leave them overnight. Okay, and so you're going to spin them down, replace that pink media with the blue media, and the blue media has the normal nitrogen. And so as the bacteria is growing in that blue one, if you go a full 20 minutes, then you're going through a replication cycle by then, and it should have made a new DNA strand, one daughter strand. And what you do is you track the weight. It shouldn't stay there, or you're wrong about the replication cycle, okay? So when you spin it, you go to that first generation, it should shift in some way. And what they saw is it didn't shift into two bands, one pink down here. Oh, we can do like the advantage of having this pen as we can. So you might have pink down there and blue is the next one, right? And you might have blue up there as one scenario. That means you made a completely new blue strand and you still had the original pink strand. That would be conservative replication. But what they saw instead is they saw a mixture where they held together somehow when they spun them, means that they were hydrogen bonded to each other. And there was an in-between weight because it was half of the heavy and half of the, when I say in between, you're going to have a control so you know what that top weight is. So this is not showing their controls, which they would have known, like here is if it's completely N14, not the heavy. And they would know with a control that this is N15, the heavy. One round of replication, and they say, oh, but it's in between both of them, must be semi-conservative. And then you can track that pattern. They're going to pull apart again. So up here's what's happening molecularly at that time in that generation number one. That's why you only have an in-between band and only one in-between band, because all the DNA went through a cycle of replication. Now, what are you going to do in generation two? You might want to draw from generation one to generation two out, because a lot of students can see generation one pretty easy. But what you're going to do is pull the blue to one side up here, pull the blue to one side here, then pull the pink and pull the pink. And you're going to generate a new blue for all of those four arrows I just put on it. So that means you're going to have two complete blues, because that was this guy up here and this guy down here. And remember, it's still in the N14, the blue lighter weight nitrogen. And so it's using that to metabolize all the nucleotides. And so that means that it's still adding a blue strand in this case when it goes through replication. But these middle ones, when you pull them apart, it started with, I already used the blue. So now you have pink as the parental. Here's the pink, here's the pink. So you have a pink strand. And the new one is still going to be blue. We still have it in 14, and so you have a mixture of pink and blue. So that means if we analyze what's going to happen in the centrifugation after 2 rounds, so this is the second generation. So if we look at what's happening in the seq profile analysis of the macromolecules, we now do have completely blue. And we have a little bit still left of this rapidly dwindling original heavy labeled N15. And so as you go through more and more generations, this will start to disappear smaller and smaller, and this will get bigger and bigger. And you lose a little bit more of the information. Most of the information was here in the first generation. So you can see it looks about half and half, just like the molecular profile up here in generation 2. But then you go to generation 3, and you still have a little bit of the pink mixed with the blue band, but it's dwindling. Okay. Okay, and then there's also the Taylor-Woods-Hughes experiment. So that was in bacteria. I mean, we're more complex. So, you know, eukaryotes, so let's use a plant because you can't mess what's in straw. So they also showed that in higher eukaryotes follow a semi-conservative replication mode by using the broadbean plant. They use the root tips. Why they use the root tips?
Why?
I'm not even a plant person, and I know this. Why do they use the root tips?
Because that's where all the replication.
Yeah, same reason you get a slide in Gen Bio, probably botany too, of tips of stuff, right? Because that's where the growth is. And at this growth, that means we're going through the replication. And they chose this particular plant because the replication time, the time to generate another cycle and go through the S phase was known. And so that was pretty important over here as well. They had to know when to look, right? And so this was known for this plant. They labeled differently. This is one that is radioactive in a way that we can see it. So the labeling in this case, we can see it by film. The labeling in the previous case, when it was labeled within 15, we saw it with the seq technique. But here we're going to see it with an autoradiogram. Oh, it's called autoradiography, but the sheet of paper that shows the exposure is called an autoradiogram. Not like a cracker. Okay. So later studies with other organisms also reach the same conclusion and support an overall picture of using semi-conservative across every organism and support the Watson and Crick theory. Okay, so looking at the Taylor-Wood-Hughes experiment, what we're looking at is you're starting with labeling replication with the tritiated thiamine. So tritiated, H3, or the word for it is tritium. So you'll say tritiate, I can't even spell it, tritiated, whatever you're talking about. Okay, so here is unlabeled, but then you grow it in the presence of tritiated precursor molecules for the cell to use to incorporate into the newly synthesized strand. And if you're only looking at the strand, you don't have to worry about other things being tritiated. They are. Other things are massively tritiated, but if you're popping the cell at metaphase, then you will see which of the chromatids are being labeled. And an unlabeled chromosome proceeds through the cell cycle in the presence of that tritiated thiamine. And as it enters mitosis, both sister chromatids of the chromosome are labeled as shown in the upper right by the autoradiogram film when you do the exposure. But after a second round of replication, in part B on the left, this time the absence of the tritiated thiamine, only one chromatid of each chromosome is expected to be surrounded by grains. So these little points, speckles, correspond to a reaction of burst of light on the autoradiogram, and they're called grains. Equal labeling. Labeled, let's say labeled instead of labeling, labeled molecule. Except, okay, so you'll see this this labeling, except we should mention where there was a crossover event between sister chromatids. And you can see that over here as well, where you have these crossover events. So you have the original, you have the new one that's labeled, but you do have to put into your theory the possibility for crossover. So, and that's what their autoradiogram shows. And you can even see it in their theory, their schematic. This is how in, what is it, 1967, did we say? When it was? 1957. So this is how you make a schematic in 1957. If you want to read their paper, just Google Taylor-Wood-Hughes. In the past, this was one of the challenge assignments. So it's not worthless. It's kind of fun to see how, and they phrase these older papers very much so like a story with, you know, let me tell you a story about us, you know, so it's kind of a fun read. It's very different than papers now. So autoradiogram, I keep saying bursts of light and film, and I say autoradiogram instead of autoradiography. Okay, so the technique is called autoradiography, and you're going to have a particular type of radioisotope that is emitting incidences of energy that, based on how you layer chemicals on the film, can be sensed by the film. And what you want to do is keep it protected from light, because light has lots of energy, and the film is exposed to light, it'll bleach it completely black. But otherwise it's clear. So if you can keep it in a dark room and only expose it to your fixed whatever you want to know about. So these chromosomes are probably prepared on a slide that doesn't move the chromosomes around, and you put the film on top. And then there's a trick where you put it in a light type box, for obvious reasons, right? But this next part's the new stuff, that has like mirrors for the energy on either side of that sandwich. And what it does is it ping pongs the energy from the tritium, or whatever radio label you're using, ping pongs it back and forth through the film, so that you can, over time, as you leave it in there, the black spot grows. So you can have something really radioactive, expose the film, take it off, and it's like, that's really hot. But tritium's not that hot. And so probably you put the film in it, you close it up, and it takes about 3 days before you can even look at that film, because tritium's not that. Some people, and there's different fields of thought with people that work with radiation. There's those that I don't like when I put a piece of film on something and then I take it off 10 seconds later and it's like bleached out, like completely black. The grain is like, this is what I've had some experiments look like. Like, say we're analyzing, and so my experiment looks like this. You can't even see the chromosome structure after 10 seconds. And that's if you were to use 32P. Okay, so 32P does that. It's really high energy, and it's real carcinogenic. You have to work behind. You work like this behind, like, protection. You protect all the good stuff, and then you don't care about your hands. My hands are going to die soon. So that's 32P. Lower energy radioisotopes like tritium, it takes like 3 days instead of 10 seconds. But you don't even work with shielding. It can't break your skin. So that's an example of the different radioisotopes that you can label. Any tritium is a H, 32P, so P. We've talked about 35S before as well. So there's different, there's iodine, iodine labeling. That's I-132 or something like that. I can't remember. But they have different levels, and you want to pay attention to what macromolecules they label and if it's specific for what you want, but also like the energy level. And so they show up as these grains. So this would be unlabeled right here, and this would be labeled. Those black dots are incidences of label, and the kind of gray background is just a back reaction of a low level of energy exposure to the film. It's convenient because you don't have to take it out and then trace where that molecule was. You can see the back energy as well.
So that bottom picture would show like crossing over events too, right?
Mm-hmm. Like there's not a crossover versus here there is a crossover, and then or not up here the first arrow there's crossover. Down here this chromosome didn't have one. Mm-hmm. And then you're going to also have grains that are floating in nowhere, so you do have to use your brain, and you have to say something like, that's an artifact because it's not on the molecule I'm looking for. So you do have some exclusion criteria for what you're looking for, and you look for like a consistency of seeing it multiple times. If you see it just like once, it might be an artifact. You don't know. So you can't draw conclusions unless you have multiple points of the reaction. Okay, so in DNA replication, some terminology that we're going to use. DNA replication begins at the ORI, the origin of replication. At the site of replication, the helix is unwound, creating the replication fork. Replication is bidirectional. It's going to proceed in both directions. There's 2 forks per bubble. You'll see it on a picture here in a little bit. And it has a terminology for what's being made. It's called the replicon, the length of DNA that's being made in each replication process. And in bacteria, it's like the whole chromosome is one replicon. But in eukaryotes, we have multiple because it's so much bigger, it needs to have multiple, or it'd take forever to replicate the chromosome. All of the chromosomes. Okay, so bacteria is simpler. We're going to start there. So bacteria have one origin of replication called the ori. And the sequence for it is going to be species specific. And so the names can vary a little bit between different organisms. So for instance, in E. coli, so ori is the broader term of the family of these sequences. In E. coli, it's called the ori-C. And the entire bacterial chromosome constitutes one replicon. That's 4.6 million base pairs for its genomic DNA. Now, if it has a plasmid, a little like an F1 plasmid that we've talked about briefly before, that's smaller. That's usually just like 4,000 base pairs, but anything that has an ori on it will go through this process. So we can sneak a little circle in there by putting an ori sequence in the middle of it. It'll also replicate whatever little sequence we put in that 4,000 plasmid. And then it's bidirectional. This is the bubble that opened up. If you look, so this was the ori. So it's going to open up right there. And it's going to start replicating. This is one replication fork, and that's the other. And so it makes this bubble that extends out. So the process in bacteria has a certain pattern based on the enzymes that are involved. There's different types of polymerases. You're going to need a polymerase. But the different polymerases perform different functions at different times. So the polymerase is just one part of the scheme that gets it going. You also have to have other enzymes. It's great to cover it in bacteria first because you still have those needed functions when you get to eukaryotes. It's just they have like 5 things to do it instead of 1. And you might think like, why does it make it more complex? You know, simpler, just having one enzyme to do the thing should be more powerful. That's a higher evolved organism. But actually, if you have like 5 things that do that one thing, it's more controllable because the cell can turn off just one of those 5 and kind of fine tooth the reaction. Okay, so we're going to pay attention to the categories of the functions as we talk about in bacteria, because the categories will apply in eukaryotes, and you need to know like what they do function-wise. You don't need to know that there's 20 and memorize the list. Okay, but in bacteria, you want the list, you want the function. Then we'll move into more complex, and you just keep in mind the overall picture. So in DNA replication, semi-conservative and bidirectional, this will require several enzymes. This was done by Arthur Kornberg and his colleagues. Roger Kornberg does transcription, but Arthur Kornberg does replication. Father-son. He just moved on to the next part of the DNA molecular biology central dogma. So Kornberg isolated DNA pol 1. It was the first of several enzymes involved in E. coli replication to be identified. And it directs synthesis of the DNA. This is a polymerase, will direct synthesis of the DNA. It doesn't have particular binding sites for A's, G's, or T's. That's the hydrogen bonding part of it. It really only has a synthesis of the bond between nucleotides and has sometimes a separate function, separate from the polymerase of proofreading. And we'll talk about that. So it requires, for DNA synthesis, it requires a DNA template. It does not synthesize a strand without having a code for that hydrogen bond to match to. So if they're not making Targoff's rules complementary connections, then there's nothing setting there for the polymerase to synthesize. So it requires a DNA template, and it requires the nucleotides to like fly in and match, right? If they're not there, you just can't build it. So please add DNTPs to your PCRs. Okay. So to elongate by the polymerase function, it occurs in the 5' to 3' direction. Let's pause right here. Anything that's synthesizing a nucleic acid that is currently known is always going to add to the 3' OH. And because we have anti-parallel rules and we have complementary basis. If you use those rules, you don't have to memorize the whole structure. You just need to know when there's an arrow growing and a polymerase is about to put something on it, that's a 3' end. Even if we get in chapter 12 when we're talking about transcription, it's still a 3' end when you're generating an RNA strand as well. So polymerases add to the 3' OH. I do not know of an enzyme that breaks this rule. It'd be really cool if one of you discover like something weird, like a virus that does something. But there's a lot characterized, and it's always a 3' OH. So just put it in your head for sure. Always added to the 3' end. The nucleotide will be added by cleaving off the 2 terminal phosphates. This is a diphosphate, and it's called an inorganic phosphate by adding a little I to it. So you might see that if you ever talked about sequencing or something like that. The inorganic phosphate is one way you could sequence. You just look when the phosphate's generated, and that means you added something. So this can be seen, the inorganic phosphate. It's also energetic. By cleaving off those high energy bonds, it fuels the process as well. So the nucleotide going in is a triphosphate, but when it's integrated into the strand, it's going to only have 1 phosphate left in there. And so DNA synthesis proceeds by addition of another nucleotide to the newly available 3' end. So every time you add a next one in, see, then this is going to be gone. Look, it lost the water, and so that oxygen's gone. It's not there anymore. You can't add like another thing right there. It's only going to go in a chain for the polymer, and it's going to add to the newly available 3' end of that nucleotide that was just added. So we need to introduce the fact that these enzymes have more than just a polymerase function. And so this is a concept that's never really talked about. I don't even know if in biochemistry, it just developed over the course of like 6 classes that I had. Eventually I was like, oh, proteins have different like subunits of them or domains that do different things. And before that point, when this epiphany just fell out of the air, because I never had a professor tell it to me, so I don't know where it came from. But you get enough of professors saying it that eventually you're like, oh, that's how it works. Okay, so I just put it in a slide. So we think of, before that point, you think of, you know, if I say polymerase, it's only going to synthesize, they're really specific. They only do one thing. That's what you learn about in Gen Bio. They can only ever recognize their substrate. Not true. And they only have one function, because that's what, no, it's not true. You can have more than one domain. A polypeptide chain, part of it can fold into the polymerase and the other part can fold into something else. And so in that way, you have reactions kind of linked to each other that helps a particular set of functionalities also be linked. So one thing about synthesizing the DNA is it needs to be kind of correct also. So often these polymerase enzymes have a polymerase domain, and they also have a proofreading domain, because that's really important for the DNA to be made, but not just be made, it needs to be mostly accurate. So in general, this is the in general, not just for polymerase, but the in general slide. Proteins have domains or subunits. They can be a different polypeptide strand that associates with each other. And those domains or subunits will have different activities based on their different shapes, because they're coded differently from different genes. So they have different amino acids, they fold differently. And it's called a domain if it's on the same polypeptide strand. It's called a subunit if it's on a different polypeptide strand, but together they're both the final protein. Even if it's a different polypeptide strand, you don't make the final quaternary structure. If you've heard that term before, we have it in Gen Bio, but it's like one slide and done. So if you're running just steam off of Gen Bio, you might have forgotten that a protein's not just one gene. Sometimes it's like 4 genes, and they associate together to form a final function. So in both cases, if it's a domain within a polypeptide strand, and it's a different domain, and then a different domain, then a different domain, they come together to form the protein. But even if it's different genes coming together, you might need different genes to come together to form the final functional protein. So a domain is a functionally defined region. Sequences within that domain are usually conserved. They have names different than the protein. So what are some domain names? There's like the WD40 domain. Oh, it's a really pretty protein. It looks like I don't know, tool names. It's like a lug nut. Looks like a flower. Looks like that. And WD40 will be in different proteins. And so it'll be like, for instance, maybe a domain needs to associate to DNA. A protein needs to associate to DNA. So they will commonly have a domain that associates, a SHT domain that associates to DNA. And then the other domain is what recognizes its sequence, though. So it'll slide along the DNA with that other domain, but then it'll recognize only its sequence with the next domain. And then maybe its activity is to cut. And so you have another domain that has the nuclease activity. What was that?
There's like something flying in my face.
Oh, okay. I just thought you had an epic question.
No.
So those domains, like the in-between parts of the polypeptide, those won't be that conserved, but the domains will be conserved. And you can even see them just looking at a gene. You can say, oh, that sequence right there, it's in the coding sequence because it's after the ATG. That's going to bind DNA because it's got a SHT domain. So I don't know what it does to the DNA, but it's binding DNA. And then you can design your experiments. You have the gene sequence, put it in a vector with restriction cloning, grow it up, and then add some DNA to it, different types of DNA, see what parameters it needs to bind so you can start to work on its function. So domains are a powerful tool because they're conserved. The sum of the domains makes up the various activities of the protein. You can have a binding domain, for instance, a localization domain. You can have a domain that tells it to go out to the cytoplasm. You can have a domain that tells it to go outside the cell. You can have a domain that tells it to go into the nucleus after it's made from the ribosome. So it's not normally in the nucleus. Go back. Go back where you came from, except for you haven't been there before, but your mama was there. Go back. Anyway, so catalysis, the lysis, the nuclease function, the whatever function, it could have a domain for that. Sometimes the domains are just to draw other subunits to it or other proteins to it. So it forms like a mega complex that's the final functional form. That's called the oligomerization domain. So that can be like, I need one polypeptide strand, but I need two of them. That would be called a dimer domain that makes it stick together. Maybe it needs 4. That's a tetramerization domain. Maybe it needs 2 different polypeptides. It's a heterodimer domain. So like that kind of thing is really important for the protein to work and can be recognized just with the sequence. Okay, so these different subunits that provide different activities and different domains are important to understand when we start talking about these proteins having different abilities. We're not talking about, like, say for DNA polymerase 1 here, it has the polymerization domain, but it also has different types of exonuclease activity. Since these are different, and now that you understand domains, and you learned back in Gen Bio, oh, a protein only has one specific reaction. That's not true, but a domain only has one specific. So the polymerization will be like one domain that does it, or subunit. In this case, it's subunits. You have another subunit that will do the 3' to 5' exonuclease. And you also have another subunit that needs to do the 5' to 3', because they have to be different. And then you have relative amounts of those polymerases in the cell, much less of polymerase 3. Polymerase 1 was studied first. But it turns out polymerase 1 is not the main guy, even though there's more of it. Okay, so for polymerase 1, 2, and 3, they all have this feature where they cannot add DNA without a primer. There might be a template, and the template's in the polymerase. But because the reaction is forming a bond between 2 nucleotides, it can't form a bond to air. And that applies to every single nucleotide setting there. It just can't add that first one, so it can't catalyze up and down the template. So you absolutely always have to have a primer for DNA polymerases. Not so for RNA polymerase when we get to chapter 12. And the way that chapter 10 gets around it when we're making a DNA strand is to steal from chapter 12 a little bit. So it cannot initiate DNA synthesis. It needs help. It needs a primer. The primer is referring to, when we say a primer, say that's an open parental strand, and these are the bases sticking out. A primer is just a short oligo that matches the first bit of DNA. And as long as you have that primer sitting there, then polymerase can go. Okay, so a primer, we're going to say it over and over again, but there, I drew it once because I don't know if we draw it later. It'll be shown with cartoon for sure, but I don't know if we show that it just needs to match like 10, 5 bases even, just needs to match a little bit. So these other activities, when we say an exonuclease, what that's doing is as the polymerase goes, it's got the 3' on this end, but say this was a mistake. It's got like a back subunit back there that recognizes it as like a chemical bump when it hits it. And it's like, whoa, 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 whoa. And it scoots back and bumps it out. So we can try again. Right? Because it's got that bump there, it needs to cleave it in the 3' to 5' direction. So this is called proofreading. And it's important for polymerases to have it, so all of them have that subunit ability. Now the 5' to 3' is something different, unique to what pol 1 does do. It's not the main guy that synthesizes the daughter strand, but what it does need to do is fix those stupid primers. And so it comes back and goes, nope, 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 and cleaves that primer out so it can be fixed back into DNA. So this one, you're going to hear about at the end of replication when we need to fix our duct tape fixing the garage, which reminds me, I need to fix other polymerases. We talked about one, it was discovered first. All the polymerases have that 3' to 5' back it up and fix it subunit. So this is the proofreading and repair. Pol 3 is responsible, he's the main guy. He's responsible for polymerization, essential in vivo. How they found this out is they knocked out pol 1 after Arthur did all this. So, oh, look at what we found. It's great. We know how it's working. They knocked it out. Remember, we talked about lethal mutations and stuff like that. And we'll talk about it again here when you talk about how to handle lethal mutations in this chapter. But they knocked it out, and those cells were okay. They're fine. I mean, they didn't do great, but they were like, no, we're okay. So how are they growing and dividing without the polymerase? It's because pol 1 is not it. Okay, pol 3 is the main synthesizer. Pol 1 is responsible for removing primer. The primers that get put in are, remember I said they steal from chapter 12 for this trick. So they're RNA. You don't have in your chromosome these little pieces of RNA just sitting around. Okay, so that implies something had to fix it, and that is pol 1. Pol 1 goes in and cleaves it out. Okay, and the synthesis will fill in that gap and fix what it took out. And they'll fix it with DNA. And so then you have a complete strand that's DNA. And then pol 2, 4, and 5 in order of discovery, that's they participate in various aspects of repair of the DNA. And so they're sensing mismatches between the strands, because when you have a damage, UV light damage, you have a transition or a transversion. You probably, do y'all talk about transitions, transversions? The words TNT, the dynamite. Literally, they're breaking one nucleotide in the base pair, but not both at the same time. So the cell can see that it's not matching, like something's wrong. And so these polymerases go in and cleave them out and try and repair. Okay, and it doesn't always work, but okay. So when we have a lot of things coming together, like what do we call protein versus enzyme versus domain versus subunit? When you have everything together, you call it a holoenzyme. So a oleoenzyme is a term for assembly of all the subunits needed for a functional protein. You would just say, can't we just call it a protein? The problem is, like I was just teaching the entire section of proteins. One gene makes one protein, you know, like, but it's not really that way. So you really need a different term to make people think that way. So oleoenzyme is a term for everything that comes together for a particular function. So the oleoenzyme for pol-3 has the core enzyme complexes that synthesize the DNA, made up of subunits for polymerizing and proofreading. So everything that you need not to destroy your organism. And those are here. That's the core. Alpha, epsilon, sigma. Unless I have my Roman stuff messed up, in which case you could just correct me. Because I can draw it out. It like words, Dr. Howard doesn't work good. So I might get the Roman numerals mixed up on you. Just let me know. You Greek people, jump in. Anyway, so subunits related to handling the DNA strands while synthesizing are down here. And that will have the gamma complex that has like how you help load the DNA into the polymerase. Like it's not just like, oh, here's a template strand and the core enzyme sits there and it's like everything's happy. No, the DNA has all these forces on it related to pulling apart the strands, keeping them apart, loading it. Well, the polymerase has these loading functions. And a sliding clamp that helps it stay on the DNA. You don't want it to fall off in the middle of the strand and make a half strand. And those are called processivity, keeping it running. Okay, we're going to have the oleoenzyme when we start up next.